schizogony again and again. Now, after the schizogony in the erythrocytic stage, some merozoites, after invading the RBCs, they can form the male gametocyte and the female gametocyte. We call this stage is the gametocyte forming stage. Okay, so in human being, 3 stages. One is in the liver cells. The other 2, both of them are in the RBCs. Okay. Now here is a mosquito. He will bite you and sucking the blood. Now, what parasites stages can be sucked by the mosquito? We said in RBCs. In RBCs, how many stages can we find? In RBCs, how many stages can we find? Yeah, could be the ring form, could be the latitrophyte, could be the schizoid, the immature and the mature schizoid, and male and female gametocyte. Okay, so now all of these stages will be sucked by mosquito, but these asexual stages, all of them will be killed by mosquito, and only female and male gametocyte can survive in mosquito. In mosquito, at first, in the mosquito's stomach lumen, the male gametocyte, the female gametocyte can form the male gamete and the female gamete. Then they are fertilized to form the zygote. The zygote can develop the ocnid, can develop the ocnid. So here, each ocnid can form one oocyst. So here, this is the mosquito stomach. On the surface of the mosquito stomach, we can find there are a lot of oocysts. There are a lot of oocysts. So here is not in the stomach lumen because the banana-shaped zygote can pass through the stomach wall, and under the stomach wall can form the ocnid and form the oocyst. Then in the oocyst, they need to undergo the sporogony to form the sporozoites. At last, these sporozoites will wait in the mosquito's mouth and then will be injected to human being. So in mosquito, there are 2 different. One is gametogony in mosquito stomach lumen. One is sporogony. Sporogony is in the mosquito's wall. So here, because this one is a sexual reproduction, so mosquito is there in a definitive host. So here, so this picture this picture shows the life cycle in human being. So let's review the human being, the life cycle in human being again. This one is a sporozoites. Invade at first. What cell here? Could you tell me at first the sporozoites? What cell can the sporozoites invade? Yes, good, liver cell. In the liver cell. So at last, look, the liver cell will be ruptured. So many merozoites are released into the bloodstream. Okay, now they can invade RBCs. At first, look here. They are very small. Do you think they are the ruby ring? Okay, so here is a ring form. Then ring form become a little bigger. It's a latitrophyte. Latitrophyte can mature. They can undergo the schizogony. At last, a lot of merozoites. How many? Each ring form can form 12 to 24 merozoites. Yes, good. Then this RBC will be ruptured. So here, merozoites can invade another. So here is a cycle. It's the erythrocytic stage. But besides this erythrocytic stage, okay, so the merozoites can form here is the mature and in the male and the female gametocyte. So this is a gametocyte for plasmid for falciparum. This is 2 for vivax. So here is life cycle in human. Okay, now the review for the human extra-erythrocytic stage for tachysporozoites at death, for bradysporozoites 6 to 12 months. Then erythrocytic is average is 48 hours, resulting more merozoites and gametocyte forming stage. So 3 stages. So here, let's show some pictures to you. This one is schizoid in liver, the schizoid in liver. Now, we said in the liver cells, both in the liver cells and in RBCs, we can find the plasmodium. But what cells are easier for us to find the plasmodium? Liver cells or RBCs? Which one is more easy for us to find the plasmodium?
BC.
RBC is good because we take blood is more easier to than to take the liver cells. Good. So we can do the blood smear and we can use the right staining or the Giemsa staining to stain the blood smear to find the plasmodium. So this is a RBC with plasmodium in it. So after staining, RBC is still red. Then the nucleus, we know that the RBC hasn't nucleus. What about the plasmodium? The plasmodium nucleus is red or purple, and the cytoplasm is blue. They are blue. And also we can find there are some malaria pigment that are brownish black. Okay. So brownish black. And sometimes we can find some dark red in the RBCs. It's an abnormal RBC. We call them Schaffner's dots. So if we want to find plasmodium in RBCs, we need to stain the blood smear by using right stain or Giemsa stain. Now let's see the erythrocyte size here. Let's see. Here is a merozoite. Okay, now this is a merozoite is invading RBCs. Okay, so after staining, look here. Did you see? What about the blue things?
Nucleus.
Blue things.
Cytoplasm.
Yes, cytoplasm, the plasmodium cytoplasm, right? And what about here, the red dot?
Nucleus.
Yes, nucleus. So do you think they are similar with a ruby ring? Okay, so we call them the ring form. Now, see, this is a electron microscope picture. So can you see some RBCs are different from the normal?
Yes.
Do you think this one is normal?
No.
What about this?
Abnormal.
And this?
Abnormal.
Yes. But it seems this one is normal, right? These two. Okay. So what's abnormal here? We can find that there is a hole, right, on the RBCs. And there are some balls on the RBCs, right? So these RBCs have the different shape from the normal RBCs. So how can the merozoites invade RBCs? Because there are some receptors and the cell ligand on the plasmodium. So when the ligand of the plasmodium capture to the receptor on the RBCs, then they can invade these RBCs. So if there is no receptor on RBCs, it means what? So could the merozoites can invade RBCs and cause infection? If there is no receptor on the RBC's surface, so can the merozoites adhere to the RBCs and invade these RBCs?
No.
No. It means these RBCs cannot be infected by this plasmodium, right? And it means these people won't be infected. And what about the receptors on the RBCs? It's a blood type antigen called definitis antigen. And some people haven't this antigen. So these people are unsusceptible to Plasmodium vivax. Clear? In West Africa, about 90% Black people haven't this antigen. So they won't be infected by Plasmodium vivax, but they are susceptible to falciparum too, because falciparum and vivax need different receptors on the RBCs. Okay, so this is decided by genus. Now, let's see the development of the ring form. So we can find that they are becoming bigger and bigger. Okay, so we can find here the blue cytoplasm are bigger than the ring form. And also we can find that there are some sealed pods on it. So we call them lateral trophozoite. And we also call these stages amoebic trophozoites because it has a sealed pod since. Now, and do you notice the RBCs are different? RBCs is different. What are differences between the RBCs here?
It got Schaffner's dot.
Less hemoglobin.
Good, very good. A little bigger, and the color is lighter, and we can find the red dots. They are Schaffner's dots. So just like this picture, we can find that these RBCs are abnormal from the others, Okay. Now, the lateral trophozoite develop again. Develop. Okay, so we can find that here. Okay, they were divided into pieces. Okay, so the nucleus divide first, but the cytoplasm won't divide now. So this is an immature schizont. Then after the nucleus have been divided into 12 to 24 pieces, okay, so the cytoplasm can divide into the same number of pieces, and each piece of cytoplasm can around one piece of nucleus, so form the merozoites. So here, with merozoites is a mature schizont. Now the schizont mature, so the RBCs will be ruptured. So here is the schizoid, and the features is cyclicity. Now, besides this asexual reproduction, they also undergo the microgametocyte. This is a male because the male gametocyte is smaller than the female. So the male gametocyte is also called microgametocyte. and the female gametocyte is also called macrogametocyte. Okay, here. So in the PV patient, PV infected patient, in their RBCs, we can find so many stages. We can find so many stages. Now, what about in the mosquitoes? Look, in the mosquitoes here, male gametocyte, female gametocyte, that can form the male gamete, female gamete, then fertilized to form the zygote here. Zygote is round, that can form the banana-shaped opening. So opening can pass through the stomach wall. Then under the stomach wall can form the oocyst. In the oocyst, look, so many sporozoites are formed. At last, they can move to the mosquito's mouth. So this one is a male gamete, not gametocyte, male gamete. Then this one is banana-shaped ocinate. Then this one is the mosquito stomach, and under this mosquito stomach we can find it here the round oocyst. So look, the round oocysts. Now here is a mosquito is biting human skin. Now the characteristics of life cycle here: intermediate host human, definitive host is mosquito, infective stage is sporozoite. They are formed in mosquito, and infective mode, mosquito bite skin of human. Here, let's think about this question: organ transplantation or blood transfusion. Now let's see this picture. Okay, now let's see picture. What about blood transfusion? If this blood from the donor haven't been examined completely, maybe perhaps there are plasmodium in the RBCs. So we said, how many stages can we find in the donor's blood RBCs? So many. And all of the plasmodium stages are injected to the receivers. Can this plasmodium continue development in receivers? Could you tell me? Yes. If this plasmodium from the donor can continue development in receivers, so it means that blood transfusion can transmit plasmodium, right? Okay, so we expose here. The RBCs are from donor. Then there are plasmodium in it. If these RBCs survive in the receivers, okay, so this plasmodium can also survive in the receivers. They can finish their schizogeny in donors' RBCs. So at last, these donors' RBCs will be ruptured and these mesozoites are released from the donors' RBCs. If there are definite antigen on the receiver's RBCs, okay, now these mesozoites can invade RBCs, whether it is from the donors or from the receivers. So they can invade the RBCs and undergo the schizogeny in RBCs again. So here we should know that blood transfusion can also transmit the disease here. So this is also the third disease that can be transmitted by the blood transfusion. What about the others we learned? Did you remember? What about
Organ
the others?
transplantation.
Which one? I didn't get you.
Yes, Laushin.
Which one? What are the two others protozoa also can be transmitted by the blood transfusion? What are they?
Toxoplasma gondii.
Good. Toxoplasma gondii. Toxoplasma gondii. What cells can be infected by Toxoplasma gondii? In blood.
Macrophages.
Macrophages. All of the white blood cells because they are nucleated cells, right?
Yes.
So what about the others? Could you remember?
Leishmania donovani.
Very good, very good. Leishmania donovani. And which cell does the Leishmania donovani live?
Macrophage.
Yes, good.
Monocyte.
Yes, only monocytes or macrophages. Good. But here, we said RBCs can be invaded by Plasmodium, just opposite to Toxoplasma. Toxoplasmodium can live in cells except all of the cells except RBCs, right? But Plasmodium just live in RBCs. So this is the third disease that could be transmitted by blood transfusion. Okay, so remember it. Now let's show the pictures again. So, now let's show the other pictures. So here is this is a normal red cell. This is the normal. This is RBCs. RBCs are infected with PV viruses. So here, one trophozoite, one ring form, but here there are two ring form. But two ring form can are seldom less than the falciparum. Now, this is here. Rings in two slightly enlarged RBCs. This blood smear is from 17 years old with a relapse due to Plasmodium virus. It's also another ring form, but it is a later ring form. It's a little bigger than this one. Now here, double infection. There are two ring forms in one RBCs. Then see here. Here is a later trophozoite because it has sealed pod. Then it's another later trophozoite, and we can find that this RBC is bigger than the normal. Now, and in seeing here, we can find a mature schizoid. Mature schizoid means there are merozoites, so we enlarge it. So here we can find in these RBCs, there are merozoites in it, so it's the mature schizoid. This one is immature schizoid because the cytoplasm hasn't been divided into pieces. Here, mature PV macrophages, a macrogametocyte, and this is a mature microgametocyte. Now, here we introduce the PV life cycle and their morphology in the RBCs. And later we will introduce the differences between the PV and the PF morphology and life cycle. But we will stop here. I think there are too much for you to remember. And we will introduce these differences at the next time, okay? Okay, so we finish here. See you next time. Okay.
Thank you, Laushin.
Okay, you're welcome.
Thank you, Laushin.
Bye.